what Thomas already referred to, much of what I'm going to tell you about is sort of a long-standing collaboration with Trevor Bedford in his lab, which started actually at KATP in Santa Barbara very innocently in 2014, when we sort of reminisced on, wouldn't it be great if one had some kind of always up-to-date real-time phylogenetics available on the web. And this is sort of what seeded NextStrain in the 5.5 or 5 years that followed. So there's a lot of people involved in NextStrain now that I in particular want to sort of call out these names down here, which have done a lot of the heavy lifting in the recent months. So that would be James Hadfield, Emma Hotcroft, Ivan Aksamentov in my lab, Maura Zuber, who's done a lot of the, every day sort of keeping it up to date, and Tom Sibley. I also just upfront want to call out that we are analyzing data that other people generate and share, and this is very important to say upfront. There's hundreds to thousands of many thousands, in fact, many scientists around the globe, doctors, nurses, everybody involved in this response that care for patients, generate, take samples, and these samples eventually, might end up in the sequencing lab, get sequenced and submitted. And when they're submitted, they're mostly submitted to GISAID, a database that has sort of jumped into the void. They usually do influenza work, but jumped into this and kind of enabled data sharing from all over the globe and data curation from the very beginning. So yeah, all the data that we've been using in this talk will have been contributed to GISAID and curated by GISAID. So where did this all start? And Thomas just said that COVID-19 sort of started in March, but the first time it sort of really appeared on the horizon was late last year or very early this year, when there were reports from a mystery virus causing severe pneumonia in Wuhan. And it's not that long ago, it's just about 6 months ago, where there was a total of 44 cases. So we've upwards of 60,000 cases in the US today per day, 11 of which were considered severe, right? And you have these kind of news headlines every so often, right? There's a new bird flu here, and so this this is cause for concern, but it wasn't obvious at the time that this is going to be that bad. So let me just give you sort of a bit of a history. So this press headline that I just flashed was sort of triggered by sort of 27 and then sort of later 40-something cases of pneumonia connected to a fish market in Wuhan. And then things started to happen very quickly. Just a few days later, so that market was closed. Only 2 weeks later, we had the entire genome of this virus sequenced and shared publicly. So this is remarkable, right? So about 2 weeks between the first notification of the disease. 2 weeks after the notification of this disease, we had a new pathogen, a 30 kilobase RNA virus that we didn't know before, completely sequenced and shared with scientists across the globe, so that that diagnostics and stuff could be developed. Only a few days later, there were sort of several cases that showed up across Asia, and a little bit later than in the US and in Europe. So, but at that time, life in the West was still sort of completely normal, and one sort of looked at China from the distance, very tensed of what is going to happen. But in fact, by early February, it was pretty obvious that the virus is in most places, and that a large-scale pandemic was on the horizon. So what are we dealing with? So everybody knows now this is a coronavirus. This coronavirus, in fact, has sort of a bunch of relatives, and a few of them are shown here on this sort of very, very broad scale phylogenetic tree. The new coronavirus, SARS-CoV-2, is down here, the red guy. And it's a relative of SARS-CoV-1, so the virus that caused the SARS outbreak in 2003, 2004. But it's also a relative of other coronaviruses, for example, like the Middle Eastern respiratory syndrome coronavirus, which mainly lives in camels and spills over to humans every once in a while. And then there's two other viruses here, which are sort of human coronaviruses with abbreviation, and they cause common colds every winter. And I'll tell you a little bit more about them later. They are sort of typically very mild and inconsequential infections. Now let's zoom into this, this clade here that contains the SARS-like viruses. So we have the novel coronavirus here, SARS-CoV-2. And as you see, these in this phylogenetic tree, these genomes here, these different samples, are all sort of right smack on top of each other, right? So there's, compared to the distance between these two SARS variants, the diversity within them is minute. Up here, yeah, that's sort of the old SARS virus, and in blue, you see sort of various SARS-like viruses which are sampled in animals. And there is sort of this bat isolate here, ARTG13, and then there's various isolates from pangolins, and this bat virus is still the closest relatives that I know of, with a sequence identity of about 96-something%. So just a quick sort of intermittent summary. So yeah, SARS-CoV-2 is a beta coronavirus in the same family as SARS-CoV-1, MERS, and 2 seasonal coronaviruses. The latter cause mild disease, the former severe. There's many relatives, so it's a very diverse family that infects lots of different animals. With a close relative found in bats that is about 96% identical. And I'll show you later, sort of given the rate of evolution of this virus, these 96% identity corresponds to 2-3 decades to a common ancestor. So it's actually a fairly close virus in sort of broad, long time scale terms. So what is special about this SARS-CoV-2 virus is that it seems to spread much more easily than SARS-CoV-1 or MERS. And by more easily here, I mean that it has a lot of pre-symptomatic transmission that makes it hard to control. While in many other respiratory diseases, for example, SARS-CoV-1, once somebody has it, they're immediately seriously sick enough that it's easier to isolate. But with this virus here, we have a lot of pre-symptomatic transmission, which makes it hard to sort of clamp down transmission. Okay. Now going into the diversity of SARS-CoV-2 in particular. So here you see a bit of a sketch of the genome. It is a very long RNA virus. It has almost 30 kilobases of genome, one of the longest we know of. And the first 2/3 are sort of the replication machinery. And then you have the spike protein which sits here, and a whole bunch of little proteins, some of which have rather poorly characterized function at the 3' end. While it spreads, it accumulates about 2 mutations per month, and I'll show you a little more about that later. So yeah, the main thing that I want to tell you about today is how we are using NextStrain, the platform that Thomas already alluded to track SARS-CoV-2 diversity. And some of you might know NextStrain. I'll just give you a quick overview here. So if you go on nextstrain.org, you'll see this website here. And currently it is all focused on the novel coronavirus. And I'll show you more about how we use it to track that later. But it actually started with a lot of other pathogens, in particular seasonal influenza, Zika, Ebola, where's Ebola? Here's Ebola, enterovirus, for example. It was meant as a platform to study a variety of viral diseases and to some extent also bacterial or fungal diseases via sort of real-time phylogenetics. So back to the talk. So at the very beginning of the outbreak, so this is data that we had, towards the end of January, there were just a handful of genomes. So this is like 20, 30 genomes that we see here. And I contrasted these two different views here next to each other. On the left, you see a phylogenetic tree. It doesn't really look like much like a tree at this point, because these genomes are largely identical or differ maybe from each other by 2, 3, sometimes 4 mutations. So here at the very left, you have genomes which have which are all identical, and they're all identical to this prototypical Wuhan strain. And then there's a few that are actually just one mutation removed. Here's one cluster of 2 which share mutation. And here's sort of a little group that sticks out a little bit. Again, sort of a family cluster here of 3 genomes which are almost identical. But by and large, these early genomes were almost identical, which has sort of one important lesson for us, meaning that they come from a single zoonotic event, right? It's not that we are sort of repeatedly sampling into a deep animal reservoir, but we are dealing with a single zoonosis, a single spillover from an animal reservoir to humans. On the right, you see the same tree, but now with each of these tips here placed on a time axis. And there's very little data here to infer the structure, the topology of this tree. You just see that sort of things that share mutation group together. But I will be hopping back and forth between a view where you have mutations on the x-axis versus time on the x-axis in the following graphs. Now, as I said, this was sort of early January, and there were sort of some very important take-homes at the time. There's a single source. The common ancestor of all samples likely was somewhere in November or early December. So this is not something that has been going around silently for ages. These genomes were so similar that they must have had a common ancestor in the very recent past. And other than a few family clusters, there were basically no identical genomes. Now, the situation has changed dramatically since. So end of January was about here. What this graph shows is the number of samples available in GIS8. And in fact, what you see here in pink is the collection date of these samples, and what you see in turquoise is the time at which these samples were deposited in the database. And pretty often you have samples, so the lag between collection of the sample and public sharing of the data is only like a week or 2 weeks. So this has really been sort of a tremendous effort in almost real-time data sharing, followed by sort of real-time, often multiple times a day, reanalysis of all available data. And the amount of data has been growing in a staggering way, to the extent that we now have almost 65,000 complete genomes of this virus. So this is sort of a snapshot here from yesterday, but what I'll do, I'll sort of switch again to the live Nextstrain site and show you around a little bit. So this is, if you currently go on nextstrain.org/ANCOV, you'll see a picture like this. We have here on the left, we have a phylogenetic tree. And in this case, it's colored by clades. This is sort of large scale phylogenetic divisions that we have come up with. And on the right, you see where these samples are distributed on the globe. And it is pretty impressive that just 6 months into the pandemic, we have sampled and sequenced this virus pretty much in every corner of the planet. And you already see some patterns. Sort of Asia, for example, this particular China is mostly these blue and turquoise colors, which are the clades 19A, 19B, while Europe, South America, Africa are pretty much dominated by these novel clades, sort of 20A, 20B, and 20C. Now in Nexttrain, we can now sort of change the way this the tree is displayed. So now sort of changes to the so-called sort of clock display here, where we now have on the x-axis time as before, and on the y-axis the number of mutations by which these viruses differ from these archetypical Wuhan sequence. And what you see here as a sort of a black regression line is just sort of an approximation of how rapidly these viruses pick up mutations as they transmit from one person to the next. And this is pretty much, this year comes out to be sort of 24.5 substitutions per year, meaning about 2 substitutions per month, or 1 substitution every 2 weeks. So that is sort of the pace at which this at which this coronavirus picks up mutations as it spreads along. And these mutations, for the most part, don't really have a measurable effect on the virus itself, but they can help to track how the virus is spreading across the globe. And this is something that I'll go into a little bit more detail. Sort of going back to the standard way to display the tree, I can now sort of look at sort of various other features of the genetic diversity. And one aspect, for example, that we have down here, we have this panel of the genome that I showed you earlier. And each of these little spikes here, they indicate positions in the genome that have a lot of diversity. In fact, they show you sort of alignment entropy at a particular position in the genome. And you can, for example, click on one of them, sometimes a little hard to find. If we click on this particular spike here, we now have the tree sort of colored by the amino acid variant at this position 614 in the spike protein. And this toggled from a D to a G. And this mutation actually happened on this branch here, where you see in this little pop-up then bouncing over a branch that there is a mutation that changed from D to G. This mutation has sort of arisen at some point in January and has since spread quite dramatically. Down here, you see, at least on some of the renditions of NextStrain, you see how the frequency of that mutation has changed over time. And it is currently sort of in our global sample at about 90% frequency. So going back to my slides, I can skip over that because that I did. So this is to say that this sort of clade 20A, along with its daughter clades 20B and 20C, have essentially taken over. And with these clades, several mutations, including this D614G mutation that I have just shown you, have become very prevalent at currently about 90%. There are reports that this D614G mutation increases transmission of the virus. In my view, the evidence for this is not exceedingly strong. My still favorite hypothesis is that the spread of this variant is explained by sort of multiple founder effects. This variant happened to have seeded the outbreak or dominated the outbreak in Europe. And with sort of a rapidly spreading pathogens, just getting into a particular place about a week or two earlier than competing with other variants can mean that you have an exponentially large head start over those. And this, if it gets sort of amplified again by dominance of an outbreak, say at the East Coast of the US, you can easily end up with such a huge dominance and rapid takeover purely by chance. Overall, there's still sort of no very strong signal that the virus has changed by mutation in meaningful ways. For the most part, we don't seem to see that, that there are changes in virulence, transmissibility, or things like that. So most of these effects are, again, just explainable by chance. But what these mutations are useful for is to reconstruct how the virus spreads and what kind of clusters are co-circulating in the community. I just wanted to sort of explain a little bit how this works. So just as a little cartoon here, I present this sketch of a transmission process where each of the red dots would be sort of an infection of one person. And each of these branch points here, they are essentially the transmission tree, which links one infection to another. So as the virus gets transmitted, it replicates, and every once in a while, it will sort of mutate some part of this genome simply by sort of making a little copying mistake. And then there's a few cases, often only a very small fraction of all cases, that get sampled and sequenced, right? And these are these sort of bluish dots here, which I labeled with letters. Now, if you now take the genomes that are from these sampled cases, here, sort of the black line is the background genomes, the diamonds are the colored mutations corresponding to these guys. You can now, by virtue of these mutations, construct a phylogenetic tree and group patients or group samples that are likely close together in the transmission tree. Remember that, in most cases, without sequencing, you don't actually observe this transmission tree, right? The only thing that you observe is case numbers. So you know that there were 50 cases in week 1 and 150 cases in week 2 and so forth, right? But you don't actually know how these different cases are connected. But this is what sort of genomics can give you, in particular over distances which are over time intervals, which are larger than 2 to 4 weeks, because that's sort of the time it takes to pick up a mutation. But in this particular hypothetical example here, for example, we would be able to say that yes, A and B likely belong to the same sort of transmission cluster because they share a mutation that is otherwise absent in the data set. And D, despite sort of being sampled right around the same time as A, is clearly in a different cluster because it has one mutation that A doesn't have and lacks a mutation that A has. So they are clearly on sort of separate branches of this tree. They're not part of the same cluster. And in that sense, this genomic analysis can sort of add another dimension, another layer to contact tracing, right? Contact tracing works by sort of recalling whom were you in contact with, whom did you meet, whom did you have lunch with, and then testing those people. This works on very small time scale and in very sort of local environments. This genomic linking into clusters works on larger time scales of the order of sort of 2 to 4 weeks. And in particular, can often exclude that there is a link between two cases. In that sense, this is a very complementary and in many cases very information-rich way of enhancing contact tracing. And one example, sort of already 2 months old now, where this is sort of very nicely illustrated, is in a large sample of sequences from Iceland, where they basically, after these winter skiing holidays, after many Icelandic people came back to Iceland, more COVID cases were found on Iceland, and almost all of them were sequenced by a collaboration that involved decode. And this showed, for once, so what you see here in this tree, all the Icelandic samples are highlighted with sort of the color code being time. The x-axis mutations, so divergence. You see that they basically completely sampled the global tree of COVID-19, right? So pretty much every major clade of this tree was brought back to Iceland by tourists, either Icelandic tourists coming back or tourists going to Iceland. Only that's probably not so much a thing in February or March. Yeah, and sort of this particular example here, for example, here I'm zooming in on the left side, and the 3 dots that you see here actually correspond to these 5 dots over here. There's a cluster of Icelandic sequences, which is nested in this case in a clade, which has a lot of other sequences from the Americas, the US, Utah, Minnesota, British Columbia, and so forth. And in fact, this group of sequences was linked to a family returning from the US recently. So this is likely sort of a re-import or an import into Iceland from the Americas, and genomic sequences clearly set it aside from all sorts of other circulation in Iceland. Now, a little bit more about sort of how NextStrain has operated in this, because NextStrain was kind of made for this kind of a scenario, right? We wanted to have a real-time tracking infrastructure in place for diseases that are around us all the time, but also new outbreaks. And we've had this before with Zika and Ebola and things like that. And initially, this worked very well. We sort of provided a comprehensive global analysis of all available data which we kept up to date and often updated multiple times a day by if new sequences arrived, and data was curated and gathered by GIS8, and up until sort of March, April, this is something we sort of could keep up with. Now we have a lot more sequence data. The global picture doesn't really change that much anymore, but what is of primary interest are sort of local analyses, like a particular US state or particular country in Europe wants to keep track of what's going on, how are these different cases connected, what kind of clusters do you have, and so forth. And this meant that we can't run these analyses for everybody, so we are sort of trying to make it as easy as possible to get these public health departments, sort of different local sort of bioinformatics, computational biology groups up to speed to be able to do analysis of the data that is most interesting to them. And this would typically result in a necessity that you have to subsample the sequences. You want all sequences from your region, but you also want all sort of neighboring sequences, all similar sequences to provide context. One challenge is that many of the people that work in these departments have rather little experience with command line tools. And they also often don't have access to HPC environments or sort of set up cloud infrastructure. So the large data sets often result in computational challenges. And an additional challenge is that many of them are not exactly trained to interpret phylogenetic analysis, which can result in issues as well. So what is our sort of current tool stack? So the Nextstrain pipeline is sort of a composable snake-make pipeline with sort of a whole bunch of little commands. There is a tool chain called Augur that has sort of various commands for aligning, for filtering, sort of aligning via math, building a tree using IQ tree, and then making timescale phylogenies using tree time. And then sort of various sort of more specialized phylogenetic analyses like ancestral state reconstruction and phylogeographic analyses or clade classification, plus the dedicated export step for our visualization. So this is something that is, everybody can do and put together and run their own analysis, but it is pretty, for people that are not used to that kind of analysis, it is kind of challenging. And we've spent a lot of time sort of writing tutorials and making it as easy as possible for sort of novice, non-bioinformatics folks to be able to use it as much as they can. And it's been working well, but it's sort of a steep learning curve for both us and them. So then there is the visualization component, which is called Auspice, which can easily be run locally just by spinning up a node server on your local machine. It is sort of deployed on our servers, but you can also deploy it on your own servers if you want. If you want sort of a completely customized instance, which looks as little as possible like Nexttrain, you can do that too. You can actually put your results in a specific way on GitHub, and then the nexttrain.org server will fetch them and display them for you. Or we have recently introduced a feature called Nexttrain groups where you can basically generate a login and you get a space, sort of a bucket on S3, where you can put your analysis and share whatever you've done with the world through Nexttrain. And I just wanted to show one sort of pretty neat example of this. So this is a group by Tulio Oliveira's group in South Africa. It's called the Network for Genomics Surveillance in South Africa. And they have been extremely active in sequencing the South African outbreak and making sort of accessible explanations using the Nextstrain toolchain to do this. And this is sort of their space where they put all their things. And I just wanted to show one of these analyses that they have done, because we're sort of very proud that people all over the world sort of start using this and do sort of exciting things with this infrastructure that we have put together. So one of their results, which I've just been sharing yesterday, is the following. It's sort of it's a summary of the situation in Cape Town. You see sort of a very pretty picture here of Cape Town. And so this is the so-called NextStrain narrative feature, where on the right, you have some explanation, and then you can sort of click through essentially a slide deck, which involves sort of interactive visualizations of the tree. So it's using the NextStrain front end to put it into context, give some explanations of what you see, have a very specific view with different colorings and different filterings, and you can generate a bit of a story using this interactive visualization. And yeah, I encourage you to look through those. In the beginning of the pandemic, we used them to keep the global situation up to date, but as that's not changing that much anymore, we've stopped doing this. But anyway, what I was sort of hoping to convey is that there is a growing community from public health and sort of computational biology researchers that sort of use this this tool chain in creative ways to keep on top of the local COVID outbreaks. All right, so let me just sort of, before I wrap up, so give you a little bit more of somewhat speculative and an outlook type ending of this. So what have we learned and what's next? So on the one hand, what we have clearly seen is that social distancing is very effective, and we can suppress the outbreak of COVID-19 if we want to. If we don't respond appropriately, it ends up in disaster. We have seen disasters across the globe. We've seen it in northern Italy, we've seen it in the UK, we've seen it in the US and various places, not just once. But we've also seen that a comprehensive and coordinated public health response can very effectively curb the spread of the virus. So this has worked in many Asian and many European countries that have sort of successfully suppressed the virus, even while sort of reopening the economy, reopening restaurants and so forth. And so this, I think the biggest take home is that it is completely in our hands as a society where this is going, right? If we just don't pay attention and let things sort of slip, cases will go up, and before we know, hospitals are full with COVID patients again. But with sort of a rather moderate but sensible response, we can keep the numbers in check. And one of the unknowns for the next coming months is actually, what role does seasonality of the virus have? Because certainly in much of Northern and Central Europe, the suppression of the virus curve sort of coincided with the arrival of spring, and this might have made it easier to suppress the virus than it would have otherwise been. And just to be clear, doesn't necessarily mean here that there is something inherent about temperature, UV, humidity, or whatnot that causes this seasonality. I'm mostly thinking here about indoor versus outdoor activities, right? Sort of these days in the European summer, I'll meet my friends outdoors. You meet in the park or something like that, rather than a crowded bar where you have poor ventilation and loud speaking. So why would we think that seasonality might have an effect? It certainly doesn't have as big enough effect to just prevent all transmission in summer, right? That's bullshit. So we know that this virus can spread in summer, but it might make it easier to control it in summer, and might mean that we need to be vigilant as the winter arrives. Now, we know from many other respiratory viruses that seasonality is a very, very common feature. So for example, here I've plotted to you the number of positive cases of influenza cases through the last 20 years in the US. And each of these spikes here is one influenza winter season, which come in different types. There's H1s, H3s, Bs, and so forth. You'll notice sort of one disturbance here to this regular pattern. That is the 2009 swine flu epidemic, where you sort of have a double peak and then sort of a pretty long low. And I'll zoom into that a little more now with data from the UK. So in the UK, the 2009 swine flu epidemic, it started actually in spring, early summer, sort of caused a peak in the UK in summer, and then a second peak as winter came in. That's not the first time that things like that happened. So if we look at the 2018-2019 influenza epidemic, again in the UK, it also sort of here had actually a double peak in winter. So this was a fall wave here in 2018, followed by another wave in early spring 2019. So this kind of seasonality is something that does happen with respiratory viruses, and it is to some extent something that we'd expect. Does it also happen for coronaviruses? Yes, I'll show you. So I've showed you before that these two viruses here, these human coronaviruses, are common cold viruses. And as you know, common colds are around in winter, right? They don't really bother as much in summer. So this slide here shows the incidence of common cold coronaviruses in Sweden, in Stockholm, over the last 10-ish years. And here too, we have a very pronounced pattern that these viruses are common in winter and not common in summer. And here you have the summary of this data modulo sort of over the year, sort of basically an average trajectory for each of these viruses by month. And they'll sort of kick in October, November, peak somewhere in December, January, March, February, and are very low in summer. So I think the take home that I want to make here is that most respiratory viruses, including seasonal coronaviruses, do have seasonality. And I believe that much of the forcing, the seasonal forcing here, is not so much weather and climate per se, but sort of the associated human behavior, which differs from place to place, right? In places where it is extremely hot in summer, people will be more indoors at that time. So it's not like a pure summer winter thing. It's more like, is it an outdoor conducive or an indoor conducive climate? And what this might mean for places like Central Europe is that control of the virus could be harder in September as people come back from holidays, kids go back to school, and the weather turns bad all at the same time. So we have to remain sort of very vigilant and react appropriately at the first sign that this virus is coming back. And in some places where the virus is not in control now, of course, we have to do everything to bring cases down there. That said, the absolute effect, sort of how much of an effect this has is very poorly known. And it might well be only a minor modulation. It could be a substantial effect. Current data doesn't really allow to tease those apart, mainly because there's so much confounding in the 6 months of data that we now have that it's very difficult to arrive at a clear-cut conclusion. So this brings me to the end with sort of one final slide on how this virus, this SARS-CoV-2, might sort of behave in the long-term future. It could become a seasonal virus after sort of a pandemic peak. It could sort of go through multiple years of sort of low circulation and then sort of settle into a seasonal virus, much like the common cold viruses that we have now, that sort of comes in winter seasons in temperate climates and has a more gradual year-round pattern in tropical climates. Okay, so I've already acknowledged sort of the NextStrain team, Trevor Bedford in his lab, including these people, and I'd also like to acknowledge Robert Dyrdek, Jan Albert, and again Emma Hotcroft and Valentin for their work on the seasonality of coronavirus. And with this, I'm more than happy to take questions. And thank you for your attention.
Thanks a lot, Richard. That was really spectacular. I couldn't have thought about a better intro to our session. Thanks for presenting this extremely useful resource and at the same time giving a nice introductory overview into the virus. We have just a couple of minutes for questions, so I will read some questions now, but I encourage you, Richard, to stay online and possibly answer some of the other questions then per chat.
Yeah, so are they in the Q&A or in the chat?
It's a Q&A. It's a Q&A.
Okay, yeah, I can also just, after, I can answer a few now and I can keep sort of hacking away at a few afterwards.
Let's go with the most popular one. Are there differences in mutational rates between clades?
Not that we know of. So the one thing that has to be said, so the mutation process is certainly not extremely uniform. So we do see sometimes like that 3 adjacent nucleotides change all at once. So there's some sort of more sort of multi-nucleotide mutation events. So we have a lot of overdispersion in this clock in a way, in this mutational process. But I don't think we have evidence for a different rate across clades as of now.
Okay,
so there's one clade which has sort of a 3 mutation hit at its base, which sort of puts it a little bit further removed from everybody else. But the overall rate, that there's not enough data right now to make a clear cut.
Yeah. Francis.
Hi. You mentioned at the beginning of your talk that about the openness of the data sets you've been using. But if you look at Joyce said, data sharing policy, it's actually not very open. And it's actually preventing people from sharing data downloaded from GSAID. Can you comment on that, please?
Yeah, so this is, we are well aware of that. What I meant is that it is impressive how people across the world have shared their data sort of in a very timely manner. The GSAID terms of conditions put a lot of strings on how you can reuse these data. And there is a quite contentious debate on, on the extent to which this is a feature or a problem. And having been at the center of this debate a number of times, I'd rather sort of not comment on it sort of too pointedly.
Okay, so what's a feature?
So I have certainly heard from many people, from sort of people that generate data in low income and low resource settings, that they sort of like to be able to assert ownership of, or sort of copyright of, not copyright, but sort of they are attached to this, so the small number of sequences they generate, and they are hesitant to put them on GenBank where they are simply in the public domain.
Anyway, we could debate.
So if you ask me, sort of a preferable solution would be to share these sequences with the CC BY license or something like that.
Yeah. But you're preventing a lot of tool developers from ingesting this data so they can redistribute it. And
I'm fully aware of that. And this is something that we've been struggling with as well. Okay. But I certainly have heard from people from especially low and middle income countries that are quite grateful for somebody guarding things.
GenBanking. So SPI is guarding it as well, but that's,
yeah. But it is a contentious debate, and
maybe
adjusting the GenBank data currently is also not really the option, because the metadata on GenBank is often a big mess.
I disagree.
So